we are doing, and a lot of work happens here also. The crystalline to amorphous strategies work using many equipments. But the one biggie failure which has come as an industry failure is from the patent point of view. Many of the industry launched the product Apixaban in the US thinking they are making a good non-infringing formulation. They have converted successfully a crystalline form to the amorphous form using spray drying or using other technologies. All of them has been blocked. None of them is allowed to commercialize. In fact, the whole process has gone back now to the drawing board, and we have been asked to redevelop as an industry, and people are afraid because the innovator company has proven that in the formulation, XRD is not the good technique. It is an inferior technique. So you have to look for a better technique to prove that the amorphous form is intact, and any contamination of crystalline in the dosage form is considered patent infringing. So these are the trials which are happening in our lab, whereby we are trying to first maneuverate with more sophisticated equipments to prove that we are not having any crystalline moiety. The option is we are moving towards liquid-filled hard gelatin capsule. Option is we are moving towards trial in hot melt extrusion and some other designing ideas here. Abuse deterrent, we are working on it. We have not yet started. We have a facility which deals in only these opioids controlled substance in Bangalore. We are the largest importer of these drugs, and we wanted to convert this portfolio from a conventional dosage forms of morphine, codeine, and such products into an abuse deterrent categories. A lot of opportunities here. I will not go details about it, but idea is just to give you this, that it has happened in the last 7 to 8 years. US products, major opioid categories are there, and we, as a CDMO, got the project signed off to develop them and launch into the market in the next 3 to 4 years of time. That's all from my side. This is the team which is responsible for doing it, and thank you very much for paying attention to it.
Thank you very much, Mr. Ashish Gogia. And in the interest of time, because this stage has to be prepared for the next event, so we will not invite any questions. And we would like to recognize Mr. Ashok Gogia for his contribution. On behalf of NIPER, we want to present this to you.
Thank you, sir. Thank you very much, sir. Thank you.
The next speaker is Dr. Anil Kukreja, who is Vice President, Medical Affairs and Regulatory Affairs in AstraZeneca, Bengaluru. He had completed his Bachelor of Medicine from Government College, Nagpur, and his Doctor of Medicine from Grant Medical College. He had previously worked with companies like Wockhardt Limited and Roche. He also worked as consultant for various startup companies. So with this brief introduction, we invite Dr. Anil Kukreja for his talk, please.
Thank you, respected chairpersons, for this opportunity. And I really want to start by thanking the organizers of NIPER Pharmacon, as well as OPPI, for providing this opportunity to be on this dais and speak to this august audience. My topic, of course, is very broad, and I kept, as I think I heard Dr. Kamla Rai also, I am also in the same stage where I have stopped preparing the slides. But as we know, all the professors, usually we give it to our students as well, so I gave it to one of my budding medical affairs colleague, and he put across the slides for me, and I will be acknowledging him at the end so that we really contribute and acknowledge particularly the students who are contributing for the same. I want to start this topic, I'm sure each one of us, and we have been enabled and being here today without masks is thanks to all the researchers, companies, doctors, nurses, and all the manufacturers who have contributed and brought the various vaccines to this world, and we have been able to control and come out of pandemic in a reasonably, I would say, a huge contribution and applause, and I would say kudos and salute to all the people who have really worked in this direction. Topic is related because we are talking about biologics, and vaccines is one of the biologics, and therefore I wanted to start with saluting all the people who really contributed for bringing the vaccines. I'm sure this august audience is aware and keeping track of all the headlines which have made in the recent past. And we are talking about the future of medicine. And if you see the future of medicine, it is moving from, as we also saw in the FDA approvals in the talk from Dr. Kamla Rai, that we are gradually going from chemical compounds to the biologics. And if you look at the future of medicine, we are talking about the immunotherapy, we are talking about the immune checkpoint inhibitors, we are talking about CAR T cell therapy, we are talking about the gene therapy revolution. And of course, we are talking about, can we reverse the aging by having a youthful stem cells incorporated into the patient population. So these are making the headlines, and I'm sure these are the future trends we need to be very, very watchful for. Again, with the help of the same slide, the same message, small molecules to plasma and recombinant proteins, polyclonal and monoclonal antibodies. And the journey doesn't stop here. The journey is taking, how do we make the antibody a long-acting antibody? And there are examples where long-acting antibodies have been approved not only for the management of COVID, preventing the COVID, but also for other viral diseases like RSV. Recently, in fact. Last week there was a drug which was approved. One injection instead of every monthly injection to be given to the preterm infant. And that's the revolution we are making by changing the antibody and making it as a long-acting antibody. There's also the journey which is taking antibody to be combined with the toxic products, so that what we used to hear during our college days, I actually studied when I used to hear one line about magic drug. So you take the chemotherapy and deliver smartly only to the tumor cells, rather than delivering the chemotherapy to across the body, therefore preventing all the adverse events which are related to the chemotherapy. And that's what is called as the smart way of delivering chemotherapy, and we are talking about the antibody drug conjugates. We are also talking about the product which will convert. Why we target only one target? Why not have a bispecific monoclonal antibody? Target both the targets which are responsible for disease pathology, so bispecific monoclonal antibody. And that's we are talking about the journey which continues, particularly in this biologic therapies. Again, going back to the basics, what are biologics? These are the medicines which are grown and purified from bacteria, yeast, plant or animal cells. And of course, there are diverse group of medicines which includes vaccine also, growth factors, immune modulators, monoclonal antibodies, and of course products which are derived from human blood and plasma. And these are different from the chemical molecules. We heard about the chemical molecules, but these are very, very different. And the high level differences, of course, again for the benefit of students, I think it's very important that we know what are the differences between chemical and biologic products. The biologic products are high molecular weight. These are very complex. These are from live organisms, and therefore there is a chance that there could be some protein element which may be immunogenic. So therefore, there is the possibility of immunogenicity. Therefore, for every biologic, immunogenicity studies have to be conducted. And that's very, very paramount and very important for all the biotherapeutic products. There are many critical steps in the manufacturing. There are less easily characterized because of the nature of the proteins. There are so many tertiary and folds which happen, which may differ. The basic message is the manufacturing process makes the product. And it's a heterogeneous mixture, and it's very sensitive to storage and handling conditions. So these are the large differences between, I would say, biologic. Just a diagrammatic depiction of what we are talking about. And again, I have not shown the bispecific. It will be far more further complex. I have not shown the ADC, antibody drug conjugate. It will be far more complex than what here I have shown the basic picture. These are just few examples, and I'm sure some of you would be following some of the monoclonal antibodies in different therapy areas. But these are the molecules which are approved. Started the journey with rheumatoid arthritis, some of the monoclonal antibodies. I am sure everyone knows about Humira, which is one of the largest selling monoclonal antibody. Then it went into inflammatory bowel diseases, psoriatic arthritis, then of course bronchial asthma, and of course more importantly in cancer. And there are so many biologics which are approved for the treatment of cancer. And we are talking about they are targeting different structures, different targets, and more importantly, these targets have not only shown to be beneficial, but they have transformed the care and the outcomes for the patients. And we are talking about almost curing some of the cancers which were incurable earlier with only help of chemotherapy. And that's the potential these biologics they bring. And some of the examples I have shown, PD-L1, CTLA4, I will talk about because that's also another topic which has been assigned to me. What about immunotherapy? So these are the immunotherapeutic agents which I will be talking about in the subsequent slides. Now, before we go to the immunotherapy, I think it's important for us to understand the cancer and immunity cycle. And these are the various steps. Simply, if you look at the tumor cell, the cancer cell releases the antigens. They are presented by the antigen-presenting cells, which are recognized by the T cells. And these effector T cells then infiltrate the tumor, and then they go, recognize the tumor cells, kills the tumor cells, and then again this killed tumor cells releases the antigens. So this cancer immunity cycle works on if there is a good homeostasis. If the immune system is powerful, there is the elimination of the precancerous cells. If the immune system is not that powerful, or there is a dysregulation of immune system because of various reasons, there is the possibility of equilibrium, or there is the possibility of escape where cancer progresses and then eventually the disease happens and progresses from early stage to the late stage, and it actually is harmful and disastrous for the patients. Now, coming to what are the different types of immunotherapy. There are broadly 4. Of course, there are a lot of research is happening, so therefore there are various other targets which are under investigation. But cytokines, therapeutic vaccines, adaptive T cell transfer, and checkpoint inhibitors. I'll come to each, but very high level without going into too much of details. Cytokines are, as we all know, immune cell release cytokines. So these cytokines are nothing but are used and administered from external sources which activates the immune cells, and therefore they help. It is just like antibiotics. We give when the immune system is not able to take the bacteria, you are giving antibiotics to complement that. So here the cytokines are there to complement our immune system so that you are able to tackle the cancer cells. Interleukin 2, again in renal cell cancer, is a very good example. Coming to the therapeutic vaccines, again here I will talk about Cytulucel T example, but there could be many examples of such kind of vaccines. The antigen presenting cells along with the antigen, which is of key interest, is combined and they are given again to the patient, and there are examples where they have shown to improve the survival also. Again, not very commonly used. A lot of more research has to be done to see whether how transformative such kind of therapies are for the patients. Again, coming to the adaptive T cell transfer, very interesting concept, the novel concept, and this is going to change. And we have seen a huge benefit of particularly one type of adaptive T cell transfer, which is CAR T. It is nothing but you are genetically modifying your T cells and reinfusing back to the patient, and you are able to activate the immunity against the cancer. And that something is, I would say, revolutionary. Some of the hematological malignancies, we have seen that the CAR T cell has shown kind of a complete cure of the disease, which were incurable. But again, technologically very, very complex. We all need to understand and see how best, as a scientist, the research community and all pharma companies come together. And there are examples in the market. We have seen the phase 1 readout from Indian CAR T cell through ImmunoAct. We see the IIT and the TMH coming together. Very good example of academia collaboration with the IITs. So engineering and medical is working together to come out with the CAR T cell, and I'm sure, knowing the competencies and capabilities we have, we are definitely surely going to have the CAR T cell products in the market. Currently it is generation 1, generation 2 and generation 3. We are currently in generation 2. Generation 3 is being worked upon particularly for the solid tumors. So there is this space, please watch out for this space. I'm sure the students who are interested in cell-based therapy, I think it's important that focus more on this CAR T cell and the practical applications of the same. A little bit about immune checkpoint inhibitors. This is the stimulatory pathway, whereas CTLA4 and PD1, PDL1 is the inhibitory pathway. And these pathways act in conjunction. If we look at further in the diagram, now this is the stimulatory pathway and these are the inhibitory pathways. Now this inhibits the activation of stimulatory pathway and therefore your immune system is inhibited. Some of the cancers, they have the overexpression of PDL1. So if you have an overexpression of inhibitory pathway, or if inhibitory pathway is more active, your immune system is inhibited to attack against the cancer cells. And therefore, some of these monoclonal antibodies inhibit either PDL1 or PD1 or in earlier slide CTLA4. So these inhibitory pathways, if you inhibit, then you are activating the immunity against the cancer cells, and that's what the checkpoint inhibitors and the examples I have shown, they do it. They basically inhibit the inhibitory pathways so that you are activating the immune system, so that immune system is able to take care of the cancer cells. And these are approved in various cancers, and I will just share one or two examples of how in the clinic, how for the patients. As we know, the most lethal cancer is the lung cancer. Highest mortality is lung cancer. In NSCLC, the 5-year survival rate was considered as only 5 to 10% if the patient is diagnosed in advanced disease. If the patient is diagnosed in stage 3 or earlier disease, it used to be 15 to 25%. Now, if you give immuno-oncology products, again immune therapy, your survival is increasing to almost 42%. You can see the 5-year survival rate. In post-IO, it has definitely increased to almost 42%. And that's what the nature, you are almost doubling the survival, doubling the number of patients survive 5 years. And that's the impact of immuno-oncology products. Let me take example of metastatic, late stage, because majority of our patients in India are diagnosed at a late stage. And even in the late stage, if you see, the survival has increased from 10% to almost 31%, again because of immuno-oncology products. So you are significantly able to enhance the survival outcomes for the patients. Now, what are the challenges? Immunotherapy works in not all patients. So there is a Efficacy is unpredictable. There is a difficulty to identify the marker. There is a need for more predictive markers. And that's the area of research where the entire community is working to see how best we identify the predictive markers so that we select the right patients for immuno-oncology products. Because definitely the disease is heterogeneous, there are immune-mediated adverse events, and more importantly, these are expensive products. So we need to really identify the patients very, very carefully. Now, as I said, cancer immunotherapy drugs are expensive, and most of the biologics are expensive. Why? Because these are complex. It involves high-tech manufacturing. So therefore, what is other emerging trend? The other emerging trend for biologic is the biosimilars. And if you see the biosimilars, there are 39 approved biosimilars, 22 are already launched. More importantly, if you see, there are so many authorities, worldwide authorities, like European authorities, as well as the US FDA, they have come out with a very good guidelines for approving the biosimilars. And if you see, it not only has a lot of saving for the healthcare system, but also look at the market. Huge opportunity for biosimilar market to come in the world. Even in India, there are so many such products approved. And I have, if you notice on this slide, I have not used the word biosimilar. Why I am emphasizing on that? Because although these are approved in India, at least as a researcher or as a person who knows this field, biologics and biotherapeutics, I do not have a published evidence that these are truly a biosimilar products as per the international guidelines. I do not see the PKPD studies being published, neither I see a comparative phase 3 trials being published, which are of non-inferiority or equivalence designs. And therefore, although these are approved, and that is something which I would say a message to the colleagues who are sitting here, I think this is one area. It's a huge market, but still we have to significantly elevate the levels or at least the evidence of our biosimilar products in India. Just last two slides maybe. These are the trends to watch for, and I'm sure many elegant speakers have spoken about these trends. I am not going to go in the details, but these are the trends which all of us need to watch for. There is some research which has already happened. We have seen the success of mRNA vaccines. We are seeing the success of mRNA-based therapeutics in the clinical trials. I am sure in the coming decade, there are so many mRNA products which are going to come. And similarly, epitranscomatics, then synthetic biology, I think we all spoke about it. I am sure the August speakers would have spoken about it. But these are the top 15 biopharma trends which are going to be the trends for future. Just to conclude my talk before I actually invite for collaboration with AstraZeneca, more targeted treatments are required. We need to identify better biomarkers. We need to see which are the combination therapy which will improve the outcomes for the patients. And more importantly, we have to also look at the immunopreventive strategies to prevent cancer and its recurrence. And that's the area for all of us to come together. Now, this is just an example of our biotherapeutic portfolio. Some of them are already launched in India. Some of them are going to be filed for regulatory approvals, and they will be launched in the subsequent 1 or 2 years. But these are all available globally. They are already approved, and they are transformative and changing the outcomes for patients across the world. Now, this is one of the slides which is, again, particularly for researchers who are sitting here. More from AstraZeneca as an organization, we have two opportunities for collaboration. One is A-Catalyst Network, and second is our open innovation. And here, these are the challenges which we solve together. One is, how do we improve the patient experiences and outcomes for early diagnosis of CKD? This is the area of interest. Severe asthma care and treatment, that's the area of interest. early detection of lung cancer, that's the area of interest. And of course, the MDTs, the oncology patient care platforms, reducing time to diagnosis, particularly for rare diseases, and particularly for the pharmaceutics and the biologic researchers who are sitting in the audience, we have a concept called as open innovation. And I would strongly urge all of you to go through the open innovation. In open innovation, there are 4 areas where we really invite all the collaborations, because we believe AstraZeneca alone cannot change the outcome. We need to work with the researchers and we need to advance the research together. And these are the areas where we have, so not only in preclinical research, clinical research, most importantly, idea incubators and co-solve. So currently, as we speak, we have a co-solve challenge, and we come out with the challenges at a very frequent intervals. We have a co-solve challenge from a sustainability point of view. I think I heard Dr. Kamla Rai speak about how sustainability is important. So this co-solve challenge is that how can we make our R&D systems more climate friendly. So there are challenge statements. Researchers are invited to come and contribute and solve the co-solve challenges together. With that, thank you so much, and I want to really appreciate Dr. Manu for putting the slides together. Thank you so much.
Thank you, Dr. Anil Kukreja, and we would like, on behalf of the organizers, present a memento.
Now I invite Dr. Ajit Dua, who is the Chief Executive Officer of Punjab Biotechnology Incubator in Mohali. She is an expert in food quality, safety and authenticity. She holds a MSc and PhD in microbiology and food technology from Punjab Agriculture University. She is also a technical assessor for NABL, and prior to joining PBTI, she worked with Markfed Canneries as Quality Control Manager and HACCP Chief Coordinator. With this brief introduction, I invite Dr. Ajit Dua for a talk.
Thank you, Dr. Bansal. And before starting, I would like to thank the organizers for inviting me to this gathering. In fact, I was finding myself out of place because I am with food background, but Dr. Bansal said there is a category, natural products, which overlaps food and pharma. So, this slide, I would like to explain, reason being you all are from different parts of India and you are in Punjab, Mohali, and I am from Government of Punjab side. So I would like to highlight that what Government of Punjab initiative has taken in the form of Knowledge City, which is diagonally at the back side of NIPER. National level institutes, ISER, ISB, INST, NABI, SIAB, and PBTI, they are a part of this knowledge cluster, which is full one sector has been dedicated for this. So it is around 400 acres of land. So this is a very beautiful ecosystem cluster. Why I have touched this? Because Dr. Rai was talking about ecosystem leaders requirements. So we have built up a very good ecosystem, and NIPER is very much a part of this cluster. I have been given, we are short of time, so I'll be skipping few of the slides. These are the major areas we are serving: analytical services, contextual research, advisory, upskilling, and incubation services. So these are the upcoming projects in Knowledge City by PBTI: Global Innovation Hub on 4 acres of land, Knowledge and Upskilling Center, Environment Resource Center. So we are basically working in the area of food, agriculture, water, environment, and little bit in health sector. And of course in the natural products to a little extent. So I'll skip this slide. We are accredited by different agencies, so total 15 recognitions are there. Uh, this I would highlight that we have collaborated with International Atomic Energy